Professor Finnemann, it's a pleasure to have you here today. Tell us a little bit about your laboratory and your work.
The main research focus of my lab is the interaction between photoreceptors and RPE cells in the mammalian eye. And particularly we're interested in renewal mechanisms that our eyes have evolved that maintain functionality of the retina and therefore vision for life. There is a particular specialized tissue renewal pathway that only exists in the eye and by which RPE cells ingest and digest and turn over parts of the photoreceptor, which helps photoreceptor cells and neurons to function for life. Over the last 15 years or so, we have made quite a lot of progress using candidate approaches to identify molecular mechanisms, namely genes and proteins that are essential to this pathway in the eye. And we're continuing with this kind of discovery work, one protein and one gene at a time.
So tell us about the current work that you're doing and some of the findings that are emerging from that work.
So I think one of the more surprising aspects of our recent work has been the fact that it is the support cell, the retinal pigment epithelium, RPE, that also is in part driving the photoreceptor renewal process. So outer segment renewal is not photoreceptor autonomous. Instead, RPE cells must signal and communicate with the photoreceptor to ensure proper outer segment renewal. And we are now only getting the idea by which types of molecular mechanisms this could happen. For instance, by RPE cells secreting factors that are then telling photoreceptors in turn how to renew. And that is, I would say, one of the more recent lines of research that we have that really pushes us a little bit into a different direction. Previously, it was always thought that the RPE is more or less at the receiving end. As soon as the photoreceptor makes the decision to shed and renew, the RPE cells would be available to intake the shed particle and digest it. But now we know that without the RPE signal in the first place, that will not happen. And we're very interested in exploring that to see if that also means that we can potentially manipulate RPE cells to signal better towards photoreceptors. And the reason why that is potentially interesting from a translational point of view is we have in the field a lot of methods and approaches that can manipulate RPE much better than manipulation of photoreceptors. So this could be very interesting down the road, I think.
So we're talking about an antegrade rather than a retrograde signaling.
Exactly.
So what are your next steps to explore this particular avenue?
So I think one of the important aspects for us is always to try to go back and forth between animal model and then also potential consequences for human disease. And the disease that is particularly relevant to this is AMD, where the types of support pathways that are received by the neural retina from the RPE will no longer function. And only recently are we getting a little bit of an idea what that means at the molecular level. We're now working towards understanding how these mechanisms really change with age, with the different types of pathological forms of stress that we know occur in the aging eye, namely inflammation, changes in mitochondrial function, oxidative stress, and how that impacts this beautiful molecular mechanism that serves us so well otherwise.
So in other words, you're starting to look at some of the elements of the inflammasome.
That is part of it a little bit. And also the idea is that the changes that the type of inflammatory reactions that we do now contribute to AMD, and that might be in fact in part causative, how those also derail the types of coordinated interactions between RPE and photoreceptors. Yes. And potentially identify new targets that are not the inflammasome directly, but those other pathways.
So talking about new targets, are there potential therapeutic interventions, and what points of therapeutic interventions seem most promising looking at this new model?
Yeah, that's a very good question. I think one of the things that we've also learned over the years from the type of molecular studies that we've done is that there are a lot of intricacies in the outer segment renewal process that are serving to maintain both RPE and photoreceptor function over many decades. However, if we think of a compromised and potentially early onset disease as in a middle-aged patient, for instance, we might not need all these intricacies if we could boost functionality of the RPE at some level. And therefore, ultimately, we would advocate to test the types of genes and proteins that are the furthest downstream in the mechanism to try to use those as targets as compared to attempting to recreate a whole functional mechanism out of a compromised situation.
I think that's a very elegant approach, and one that simplifies a lot of the...
Simplification, I think, with respect to therapy is definitely the name of the game.
Excellent. Well, thank you so much for this extraordinary presentation. We really appreciate your good work and your time, and we'll continue to look forward to more great things coming from your work.
Thank you so much. We're doing our best.